Pandomics allows you to complete a meta-analysis with all the gathered data sets, and this is called a target ID. We made one for our study for a deeper analysis of genes and pathways. In making our target ID, we used over 20 scores to rank target disease associations. These are omics, techs, KOLs, and finance, and they're divided into two major parts, those that are statistical and more bioinformatic models, and AI-based models. Artificial intelligence is used to combine the output of all models and produce a final ranking enriched with the actionable target hypothesis. And the list can be filtered according to certain criteria set by the researcher, including filters on accessibility of small molecules, therapeutic antibodies, novelty, safety, tissue specificity, and many more. Here is our non-filtered Pandomics target ID. So look at the top 20 genes here. These are the most famous genes in regards to glioblastoma, and these are often the most mutated genes of all. As you can see, a lot of these molecules are marked by TCLIN, which means they already have some sort of medicine against it. Then there are those with TChem, and these are the genes which are known to have small molecules but are not in clinical studies. And finally, T-bio, the genes that don't yet have small molecules, though it might be possible to make them. Our aim is to find new small molecules, which is why we used the different filters in Pandomics. And we specified that it must be possible to make small molecules for targeted genes. And next, we aren't interested in antibodies or the biologics filter, so it doesn't really matter if we can make them against this gene or not, which is why this filter is on red. And the safety must be high because it should be safe for a person to take any medicine created from these small molecules. And the novelty is set to yellow because we want it to be quite high since we're looking for new targets. We also decided to investigate several categories of druggable classes. For this trial, we chose to look specifically at kinases, as they're important mediators of the signaling cascade and determine some important factors like growth, differentiation, metabolism, and apoptosis. From this list, we chose to look at the HCK gene, which is marked by star. The two genes before it were downregulated, while we're only investigating the upregulated genes in order to make inhibitors against them in the future. HCK is involved in the regulation of signaling by CBL, and this pathway is highly activated in glioblastoma, and CBL, as well as the three non-receptor tyrosine kinases associated with it, are upregulated. This further shows HCK's importance in glioblastoma. But why did we choose HCK? Firstly, as you can see from the list, it was number 2 on the ranking, but we were also searching primarily for upregulated genes in glioblastoma, as this means it would be possible to inhibit and downregulate them in the future with small molecules, while the ones above HCK were mostly downregulated in glioblastoma. HCK also had a few previous studies conducted with it, and so there was some information in literature available, which allowed for some research on this gene. We also created box plots for this gene and using all of our chosen datasets, which showed the remarkable difference between how it was expressed in glioblastoma patients and in healthy patients. The blue box plots indicates glioblastoma patients, and the orange ones control patients. We looked deeper into the functions and background of this gene, and it's part of a kinase family, which means it is druggable. HCK is a non-receptor tyrosine kinase found in hematopoietic cells that transmit signals from cell surface receptors and plays an important role in the regulation of innate immune responses, including neutrophil, monocyte, macrophage, and mast cell functions, phagocytosis, cell survival and proliferation, cell adhesion, and migration. It is also localized outside of cells on the plasma membrane, so it will be easier to target it, which is an advantage. From the image on the right, you can see that the expression of this gene is the highest in blood cells and lymphoid tissue. However, we know from researching glioblastoma that its expression is also really high in the brain. The image on the bottom left is taken from the Human Protein Atlas, and it demonstrates that HCK expression is the highest in monocytes, neutrophils, and eosinophils, a medium amount of expression in total PBMC and myeloid DC, and the lowest expression is in T cells and memory cells. Here is an image to show how HCK signaling occurs in cancer cells. Part A demonstrates the interaction of HCK with receptor tyrosine kinases, which results in the activation of ERK, ACT, and STAT3 signaling pathways to promote cell proliferation. Part B shows direct association of HCK with BCR and AVL, and this eventually promotes cell growth and survival through the activation of ACT. In Part C, you see that HCK can bind directly to an acidic domain of GB130 and mediate ERK and ACT signaling to induce cell survival and proliferation in response to interleukin-6 and related cytokines. On this image, it's revealed that excessive HCK activation in a microenvironment enhances the recruitment of immune cells into tumors and promotes their proliferative and migratory ability. HCK is also involved in inflammatory signaling, and its expression augments the release of TNF-A, interleukin beta, interleukin-6 from macrophages. These cytokines can feed back onto tumor cells to perpetuate inflammation and sustain neoplastic transformation. Finally, HCK activation plays a key role in protosome formation in macrophages and promotes their phagocytotic, chemotactic, and matrix degradation abilities. This may enhance immune and epithelial cell invasion by facilitating large-scale degradation of the extracellular matrix. Now, we looked at a study conducted in vivo and in vitro on the expression and knockdown of HCK in glioblastoma cell culture and mouse xenografts. First, the researchers looked at the expression of HCK in the Cancer Genome Atlas, and the data showed that glioblastoma cells had a higher expression of HCK than in normal glial cells seen in Figure A. Then they also decided to check the expression of HCK in cell lines in vitro, and for this they used HEB cells, which are normal glial cells, and 3 different malignant glioma cell lines. Figure B shows RNA levels of HCK in the 3 cancer cell lines. The next step was to look at what inhibiting and overexpressing HCK would do to the cells. Inhibition of HCK suppressed cell viability and proliferation in the in vitro glioblastoma cells. Figure B on the right shows the effect of knocking down HCK on glioblastoma proliferation. Without any knockdown, the time taken to kill 90% of the cells was much higher for both glioblastoma cell lines. On the other hand, knocking down HCK caused the proliferation to be smaller. Figure D shows levels of cell viability, and with HCK knockdown, glioblastoma cells are less viable than no knockdown cells, showing that their rates of proliferation and growth are much lower without HCK being expressed. Then HCK was overexpressed, and this caused significant proliferation in the cell lines. Figure G shows a higher cell viability of the 2 cancerous cell lines when HCK is overexpressed, demonstrating that HCK really does contribute to cancer's proliferation and growth. Next, the metastatic potential and cell migration in relation to HCK expression were investigated. The researchers made a transwell matrigel assay in Figure A, which showed that glioblastoma cell lines had a higher metastatic potential than the same cell lines where HCK was knocked down. Then the researchers overexpressed HCK to look at cell migration and metastatic potential. Figure C shows a transwell matrigel assay for the 2 cell lines with no change in HCK levels and overexpressed HCK levels. Figure D demonstrates a statistical analysis of the assay. When HCK was knocked down, caspase-3 and cleaved PARP, 2 biomarkers of apoptosis, were more expressed, which shows that apoptosis is activated when HCK is downregulated. The researchers showed these results in Figure F, and from these results we can hypothesize about HCK's ability to prevent apoptosis, which is an advantage for cancer cells. In addition, the researchers discovered that some pathways linked to HCK, and these were EMT, which causes a loss of normal cell shape and order, hypoxia, and TGF-beta. After testing the association between these pathways and HCK through activating this pathway's transcription factor and N-cadherin, which is an EMT marker, they deduced that HCK might be involved in glioblastoma through the EMT pathway. In these figures, you will see details of the xenografts done by the study. Mice were injected with normal glial cells and with the same cells, but where HCK was silenced, and this is SHCK. From Figure A, we see that the tumor size in mice was silenced, with silenced HCK is much smaller than in those with non-inhibited HCK, marked by SH and C. Figure B and C show that the tumor volume in SHHCK mice was also much smaller. Then we assess the competitive landscape for HCK. There are two small molecules, both of which are in phase 2 for glioblastoma, but approved for other cancers already. And these are dasatinib and bosutinib. You might wonder why we chose HCK then, if there are already drugs made for it. Well, we did not notice that it had some drugs, but as we researched deeper, we did notice they had the drugs, but as we researched deeper, we found that both are very non-selective inhibitors. And this means that if each of the drugs has many different targets, not just HCK, which this could cause bad side effects in patients. For example, dasatinib also targets other SRC family kinases. Some of these play key roles in regulating signal transduction, like coupling with receptors and activating cytoplasmic signaling machinery. Without them, cells may start to work poorly and even die, and many patients taking dasatinib have said that they experienced neutropenia, anemia, thrombocytopenia, pleural effusion, febrile neutropenia, and fluid retention. Bosutinib targets members in the SRC family of kinases as well, though its primary targets are the BCR-ABL kinase. It is common for patients taking this drug to experience frequent diarrhea, nausea, vomiting, rashes, abdominal pain, anemia, fever, and fatigue. Selective inhibitors, though, are a better alternative, as they only target some specific genes, which causes fewer side effects in the end. So a proposed solution is to make a selective inhibitor targeted specifically at HCK, as this reduces the toxicity of chemotherapy and effect on other genes.